Hi friends, myself Bhavan Jha from Biology Guru. Today I have these topics from class 12th exam. These topics are crucial for board as well as all pre-medical exams. Today's topics for discussion are sporogenous tissue, pollen mother cell, monosporic development, scutellum, epicotyle, hypocotyle and perisperm from chapter 2. Second topic is BT cotton from chapter 12. First one is sporogenous tissue from chapter 2, that is sexual reproduction in flowering plants. Sporogenous tissue are a group of compactly arranged homogeneous cells in the center of young anther. These cells fill the whole interior of a microsporangium. Each cell of sporogenous tissue is capable of giving rise to a microspore tetrad. So, each one is a potential pollen mother cell or microspore mother cell. Microsporogenesis. It is formation of microspores from pollen mother cell through meiosis. After formation, microspores are arranged in a cluster of 4 cells called microspore tetrad. As the anther mature and dehydrate, microspores dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of anther. Pollen grains can be stored for years in liquid nitrogen at minus 196 degrees Celsius. It is called cryopreservation. In majority of flowering plants, one of the megaspore is functional while other 3 degenerate. Only the functional megaspore develops into female gametophyte or embryo sac. The formation of embryo sac from a single megaspore is called monosporic development. A typical angiosperm embryo sac at maturity is 7-celled and 8 nucleate, that is 3 antipodal cells at chelazal end, 2 synergids and 1 egg cell at micropylar end, and 1 central cell in the center. In monocotyledonous plants, the embryo has only one large and shield-shaped cotyledon. This single cotyledon of monocot is called scutellum. It is situated towards one side, that is lateral side of the embryonal axis. Epicotyle. A typical dicotyledonous embryo consists of an embryonal axis and 2 cotyledons. The portion of embryonal axis above the level of cotyledons is called epicotyle. It terminates with the plumule or stem tip. Hypocotyle. The cylindrical portion below the level of cotyledons. It terminates with the radicle or root tip. Root tip is covered with root cap. Coleorhiza. In monocot seeds, an undifferentiated sheath that encloses radicle and root cap. Coleoptile. Coleoptile is undifferentiated sheath that encloses plumule and few leaf primordia in monocot seeds. Perisperm. Perisperm is residual persistent nucellus. It occurs in beet and black pepper. Next topic is BT cotton. BT stands for Bacillus thuringiensis. BT toxin is produced by a bacterium, Bacillus thuringiensis. BT toxin gene has been cloned from bacteria and expressed in plants to provide resistance to insects without the need for insecticides. In effect, created a biopesticide. Some strains produce proteins that kill certain insects such as lepidopterans, for example armyworm and tobacco budworm, coleopterans, for example beetles, and dipterans, for example mosquitoes and flies. These protein crystals are formed during a particular phase of their growth. These crystals contain a toxic insecticidal protein. This toxin protein exists as inactive protoxins, so doesn't kill Bacillus thuringiensis. Once an insect ingests the inactive toxin, it is converted into active form due to alkaline pH of gut, which solubilize the crystals. The activated toxin now binds to the surface of midgut epithelial cells. It creates pores that cause cell swelling, lysis, and eventually death of the insect. Specific Bt toxin genes were isolated from Bacillus and incorporated into several crop plants such as cotton. The toxin is coded by a gene named CRY. CRY1AB control corn borer, CRY2AB and CRY1AC control cotton bollworm. Bt cotton is first commercial transgenic crop in India. Thank you for watching. I hope this would boost your information.